Now we will move forward our oral session. Our oral session will be given by Dr. María del Carmen Navarro. She is from research. She is a researcher and professor from Universidad Autónoma Metropolitana Iztapalapa, Mexico. Her paper title is Advancement on Domestic and Wild Sheep Embryo Cloning in Mexico, Possibilities for Sheeps.
Thank you very much for the presentation and for the invitation. Also, I will talk about the advancement on domestic and wild sheep embryo cloning in Mexico, possibilities for sheep in our country. Can you turn the slide, please? Okay, our country is a megadiverse one, and we have the responsibility for carrying that megadiversity in our country. How can we do this? The next, please. We are fifth place in plants, fourth in amphibians, second place in mammals, and first place in reptiles. There exist more than 100,000 species described in the country, and we have more than 800 reptile species, 500 mammal species, 300 amphibian, and 1,000 butterfly species, more than that in our country. So we have near the 10% of species registered in the world. We have an important place in the world within this megadiversity. Next, please. What efforts can be directed to the conservation and reproduction of endangered species? There are three, for instance. We have the ex situ conservation, the in situ conservation, and an in vitro conservation in gene banks. The first means that we have wild species at zoos. According to data from a federal attorney for environmental protection in our country, there are 95 zoos operating in Mexico, where there exist more than 20,000 animals of more than 300 wild species, such as birds, mammals, and reptiles. In the in situ conservation, we have what we call QMA, that means management units for the conservation of wildlife and hunting ranches. Mexico has more than 1,000 of these sites that represent 90% of the national territory. The humans are properties of owners, of possessors who voluntarily allocate them to the sustainable use of the wild species that live there. For the in vitro production for gene banks, we have what we call the in vitro fertilization and the somatic cell nuclear transfer. And also we have gamete, embryo, and somatic cells cryopreservation for these gene banks. In Mexico, there exists the National Genetic Resources Center, also called the 21st Century Noah's Ark, but unfortunately, this has only plants, no biological material of animals has this place. Next, please. But what is a somatic cell nuclear transfer? This is when a single somatic cell is transferred into an enucleated oocyte. Then both cells are electrically fused to form a single cell, which will be chemically activated to produce an embryo. In the figure, we have three sheep of different breeds. The first one and the third one are the same breed, and the second one is of different breed. It was a Dorset sheep, and I am going to talk about Dolly Cloning. She was cloned using an oocyte from a black-faced sheep that was enucleated by mechanical or physical stimuli, and another sheep who was used to obtain the somatic cell that we call here the mammary gland cell. Then these two cells were fused electrically and were converted into an embryo. And this embryo was transferred to a third sheep, black-faced sheep, and this one gave birth of a Dorset sheep that was the clone of the one that was used to obtain the somatic cell for the nuclear transfer. This is how embryo cloning works. The next, please. Some species have been cloned in the world, and we have domestic species and we have wild species. In domestic species, we have this sheep called Dolly, and we have also cattle, goats, leopard, pigs, murine, feline, equine such as mules and horses, and canines. And on the other hand, we have wild species such as gaur, mouflon, ferret, buffalo, gray wolf, camel. You in India have a wild water buffalo that has been cloned, and cheetahs. Next, please. Among other species, of course. For example, in wild sheep, we have this argali, ovisamon, that has been cloned using what we call interspecific cloning. This means that we use an oocyte from a domestic sheep, Ovis aries, and usually a somatic cell of the endangered species that means to be cloned. In this case, it was the argali, ovisamon. And it was possible to establish a pregnancy after the transfer of nuclear transfer embryos produced from the fusion of argali ovisamon nuclei into domestic sheep Ovis aries. Next, please. Another example is this mouflon, that is also a genetic rescue of an endangered mammal by cross-species or interspecies nuclear transfer using. In this case, they use post-mortem somatic cells. This means that the animal was already dead, but their cells were still alive and can be used to clone some embryos. Next, please. Next, please. Thank you. In the case of extinct species, we have the bucardo, that is also known as the ibex of the Pyrenees in Spain, that could be cloned, and there was a first birth of an animal cloned from this extinct subspecies, Capra pyrenaica, at Aragon, Spain. Next, please. What have we done in Mexico? In Mexico, we have cloned Creole half-blood that we call Criollo mestizo animals. That is the sheep that we have naturally in our country since when the Spanish came to conquer our country, and we could clone these embryos. We also cloned Charolais sheep embryos and Suffolk embryos, two breeds of economical importance in our country and over the world. The next one, please. There we have a picture about the different developmental stages of this mestizo or criollo sheep in Mexico, made by handmade cloning. We brought to our country this biotechnology thanks to Gabor Baita, perhaps the pronunciation of his last name, and he was able to come to our country, to our laboratory, and show us how to clone embryos. So this is the first embryo cloning we made using handmade cloning. Next, please. In the case of the Charolais, we were able to produce from 80.7 to 37.5 blastocyst. That means that we could ship this embryo development from cloned embryos to the stage where you can transfer these embryos to surrogated mothers. Next, please. And we have the picture of the embryos. Next, please. In another work, we worked with Suffolk sheep And we were working with sheep and ram, and were able to produce also blastocysts until these cloned embryos were growing in this wow system that is well of the well inside of culture dishes. Next, please. This is an endangered sheep species in our country, Ovis canadensis subspecies. We have 3 subspecies in our country at the north of our country, which are Ovis canadensis mexicana at Sonora, Ovis canadensis crednovates at Baja California, and Ovis canadensis wemsi at Baja California Sur, the south of Baja California. And this species figures at the red list of the threatened species. And also in Mexico is under special protection by our laws. And this is a species that is raised on national hunting ranches because of the tropheum. Next, please. This is what they call the tropheum. It means that you can achieve 2,000 USD for hunting a bighorn sheep in Baja California Sur. That is the cost for hunting a bighorn sheep in our country. Next, please. But also we have this big problem in our country, that is the illegal hunting. And there is a note on the newspaper where you can see that in a place in Baja California known as Tijuana, the capital of Baja California, on January of 2018, the federal attorney for environmental protection secured a hunting trophy. That means the head and antlers of the bighorn sheep and the skin also of a bighorn sheep, and made the owner available to the public ministry of the federation by not accrediting the legal origin of parts of a species in protection status. This is an animal in danger of extinction. We must remember that this is very, very important. The next, please. Next, please. How do we clone embryos in our laboratory? We take ear skin biopsies from specimens. In this case, there were 2 adult specimens of O.C. mexicana, male and female, from the Chapultepec Zoo in our Mexico City, and fibroblasts were derived in primary cultures carried to the 5th passage. It is very important, the passage, because you can reprogram the nuclei of this somatic cell to behave as an embryo once it is in the oocyte, in the enucleated oocyte, and fused with it. Then oocytes of adult O-Aries matured in vitro were also collected and enucleated manually, as Gabor Bakta showed how to make this, and we were working with these 2 type cells. The triplets formed, that are O-Aries cytoplast, O.C. mexicana Cardioplast, and O-Aries cytoplast, were fused to produce embryos by manual interspecies cloning. The zygotes formed were activated and cultured for 7 days in the wow system in cleavage and blastocyst human sequential medium. Then the cleavage rate was evaluated at day 2 and 7 of culture, and data was analyzed. Next, please. And there are some examples of how do we clone embryos. We have, for instance, in letter A, up and left, we have the oocytes. Then we must clean these oocytes from the granulosa cells. And we have the oocytes in letter B that are without these granulosa cells. And we can see the first polar body that tell us that the oocyte is mature. It is in the metaphase 2. Then we digest the zona pellucida that is covering this oocyte with pronase, an enzyme, a synthetic enzyme. And we have in the picture, letter C, the oocyte without zona pellucida. Then it is easy now, well, more or less easy to biopsy this oocyte to enucleate it. That means that we must take off the nuclei, that is the first polar body and the metaphysi plate, and we keep the cytoplasms, and we call this cytoplast. And we use it to fuse with it a fibroblast that is in the right picture, skin fibroblast, to use them as Cardioplast, that is the donor of the nuclei. Next, please. Then we fuse it using this electrical fusion of cells triplet, cytoplast, Cardioplast, cytoplast, after chemical activation of embryo segmentation and culturing wow system. And we can achieve the embryo development that is shown in the right picture. Next. Which cells can be used as Cardioplast? Embryonic cells, cumulus cells, fetal fibroblast, adult fibroblast, adipose tissue, embryonic stem cells, but fibroblasts are the most suitable cells for somatic cell nuclear transfer due to the ease with which they can be reprogrammed. Next, please. This is a picture of the ear skin sampling from an adult Ose Mexicana female at Chapultepec Zoo. We have in the left the female and on the right the fibroblast. Next, please. And these are the pictures of the male that was derived ear skin fibroblast from an adult 5 years old Ose Mexicana male from Chapultepec Zoo. Next, please. The results are as following. We had 60 female embryos and 175 male embryos. That means that we could get 15% of blastocyst in the case of the female embryos, cloned embryos, and 14% of blastocyst in the case of male embryos. That is in vitro production of Ovis canadensis mexicana female and male embryos in our laboratory. Next, please. And the pictures of it, developmental dynamics of handmade cloned, was also a work we could publish showing these dynamics of the development in this kind of embryos. Next, please. We can be sure that those were embryos because we stained them with HESH and we could see the nuclei that for us is the information we need to know that we are indeed working with embryos. Next, please. And some of our works about the cloning, the handmade cloning made in our country. Thanks to this biotechnology, it had been possible to clone some sheep, domestic and wild sheep. Next, please. The cleavage rate did not show significant differences between genders, that is 93% for female and 91% for male, nor in the proportion of embryo clones of these subspecies, female and male, reaching the blastocyst stage. We had 15 versus 14% respectively. Yet data are higher than those reported by some other authors who obtained only 2% of blastocysts from a mountain bighorn sheep, Ovis C. canadensis, another bighorn sheep from the north of our, this means the neighbor we have, the United States, through conventional interspecies cloning. They are also superior to that reported by other authors who, with the same technique, only obtained zygote clones of bighorn sheep, transferred into Armenian Mouflon females, but without achieving any births. Next, please. What is the main problem for the success of this cloning? There is the synchronization of the cell cycle of the Cardioplast and nuclear reprogramming. So works have been made to achieve this synchronization of the cell cycle and the reprogramming of the nuclei by using chemical agents, serum starvation, contact inhibition by cell confluence, and cellular passage number. We use the fifth passage of the cells in culture. And also donor age. It is very important if you take this fibroblast from fetus versus young or versus adult, it is working better with fetus and young species. Next, please. What are our conclusions and perspective? Embryo cloning allows obtaining in a first filial generation the expression of 99.99% of the genome of the cloned individual. It reduces the time and the number of daughter generations to obtain genetic purity in absorbent crosses. The mitochondrial DNA that remains in the oocyte used as cytoplast does not code for the phenotype, but for the metabolism of the respiratory chain. It is radically different from nuclear DNA in that it's circular, encoding 2 ribosomal RNAs, 22 transfer RNAs, and 13 proteins involved in oxidative phosphorylation and cellular energy production. Cloned individuals are, phenotypically speaking, identical to the parent, that is, to the individual from which the Cardioplast was obtained. The future perspective of the application of embryo cloning for Mexico are enormous. Genetic improvement of highly productive herds of any productive species of economic interest can be done. For example, we can rescue native Creole types of Mexican productive species, or we can use it for genetic improvement in Mexican livestock. We can rescue endemic species at risk of extinction, not only land mammals, but also marine mammals, like our vaquita porpoise. Next one, please. What is next? The next work we are going to treat in this intention of cloning embryos. We are working with ear skin derived from Angus and Zimbra bovine to produce handmade cloned embryos. It is a thesis of master science degree, and we are about to work with these animals. Next, please. This is our team, and that will be all for today. And the last, can you share the last slide, please? Because I always want to end my talks with a thought, and this is the thought I choose for today. The day is coming when humanity stops separating science and spirituality and learns to unite them in a single tool that allows it to admire and interpret all the beauty and wisdom implicit in the great works of creation, and perhaps now contemplate God, understand that, try to imitate that, and live in peace. Namaste. Thank you very much. That's all.
Thank you, Miss Maria. A very wonderful and so informative presentation.
Thank you.
Yeah. Anyone have any doubts or any questions regarding her presentation? You can go ahead.